Protein synthesis. Process by which the amino acids are linearly arranged into protein by the involvement messenger, transfer, ribosomal RNA and various enzymes. Components of protein synthesis are three types of RNA: amino acids, ribosomes, enzymes, DNA. Two major processes involved in protein synthesis are transcription, translation. Transcription is defined as transfer of genetic instructions from DNA to RNA. Transcription happens in the nucleus of the cell. During transcription, a strand of RNA is made that is complementary to a strand of DNA called gene. Transcription happens in 3 steps. 1. Initiation. 2. Elongation. 3. Termination. Initiation. Initiation is a beginning process of transcription. It occurs when enzyme RNA polymerase binds to a region of a gene called the promoter. This sign of the DNA to unwind, so the enzyme can read the bases in one of the DNA strand. The enzyme is ready to make a strand of RNA with complementary sequence of bases. The promoter is not part of resulting mRNA. Elongation. Elongation is the addition of nucleotides to the mRNA strand. Termination. RNA polymerase transcribes terminator. It detaches from DNA. mRNA is complete after this step. Processing RNA. The new mRNA is not yet ready for translation. At this stage, it is called pre-mRNA and it must go through more processing before it leaves the nucleus as mature mRNA. The processing may include 5-cap protect mRNA in and help in attachment of mRNA with ribosomes for translation. Splicing remove introns from the protein coating sequence of mRNA. Introns are regions that do not encode for proteins. The remaining RNA consists only of regions called exons that do codes for proteins. Editing changes some of the nucleotides in mRNA. Polyadenylation adds the tail to mRNA. The tail consists of a string of adenine bases. It signals the end of mRNA. Translation. Translation is the second part of protein synthesis. It is the process in which the genetic code in mRNA is read to make protein. After mRNA leaves the nucleus, it moves to ribosome, which consists of RNA and protein. Translation happens in the ribosome floating in the cytosol or on the ribosomes attached to the rough endoplasmic reticulum. Just as with mRNA synthesis, protein synthesis is divided into 3 phases: initiation, elongation, termination. Translation. Initiation. The small subunit binds to a site upstream of the start of mRNA. It proceeds to scan the mRNA in the 5'3" direction until it encounters the start codon org. The large subunit attaches an initiator tRNA which carries methionine MET, binds to the P site on ribosome. Translation. Elongation. The ribosomes shift one codon at a time, catalyzes each process that occur at 3 sites. With each step a charged tRNA enters the complex, the polypeptide become one amino acid, longer and unchanged tRNA departs. The ribosomes interact with other RNA molecules to make chains of amino acid called polypeptides chain. Translation termination. Translation termination occurs when the start codon UAA is encountered. When the ribosomes encounter the stop codon, and growing polypeptide is released with the help of various factors. And the ribosome subunit dissociate, and leave the mRNA. After many ribosomes completed translation, the mRNA is degraded. So the nucleotides can be reused in another transcription reaction. Summary of protein synthesis. At initiation phase, tRNA with its anticodon encounters the ORG start codon and the P site. The ribosomes translocate in the 5-3 direction, at which point the amino acids attached by peptide bond to transfer RNA in P site, can bond to the RNA in a site, creating a long amino acid chain based on codons in RNA. As the ribosomes translocate, the tRNA leaves the ribosomes through the E site, while new tRNA enters the A site in order to continue elongation the amino acid chain.